Hi, everybody. My name is Shannon Dunnigan. I am the SWMP, the System-Wide Monitoring Program Manager here at the GTM Research Reserve. This has been talked about a couple times now, so I'll tell you a little bit more about what SWMP is, and then we will kind of dive into some of the data. So the System-Wide Monitoring Program has been a cornerstone of the National Estuarine Research Reserve System since its inception in 1995. And it's this national monitoring network that uses standardized protocols and instruments to really have the goal of quantifying and tracking short-term variability and long-term change within our nation's estuaries to better understand how these ecosystems function and change over time. Providing us the capability to understand their response and resilience of these systems to natural and anthropogenic change, ultimately with the goal of informing coastal zone management. So as a standardized system, that means each reserve in the entire system uses the same protocols and instruments. And so as Nikki has mentioned, we do continuous monitoring. Each reserve is required to have a minimum of 4 water quality stations. And these stations are equipped with data sondes, which continuously collect water every 15 minutes, collect data every 15 minutes, things like temperature, salinity, dissolved oxygen. And we are also operating a weather station as well that collects on that same time interval. In addition to the continuous water quality information gathered by the data sondes, we also collect water samples monthly for nutrient, bacteria, and chlorophyll analysis. And we're required to perform diel collection, so over a 24-hour period, using an auto sampler. And we do this at our Pellicer Creek site. And all of this data, as Heidi has mentioned, and as Dr. Dix has mentioned, all of this data and the associated metadata are available to the public from the NERS Centralized Data Management Office website. So for this presentation, I really would like to go beyond the monitoring, as Nikki has said, and kind of focus on the nutrient status of our estuary using the SWMP data. We have 19 years of data. So I'll first do this by kind of orienting you to our sites, and then we'll dive into some patterns of nutrient concentrations and their chemical forms, and then follow up with an initial look at some indicators of nutrient enrichment. So with our SWMP sites, our northernmost site is Pine Island, and the majority of our stations are located in the Atlantic Intercoastal Waterway. Pine Island, again, is our northernmost site located in the Tolomado River. Then we have the San Sebastian station that Nikki has mentioned previously, and it's just on the north side of the 312 bridge for those of you familiar with the area. We also have a site near the Fort Matanzas National Monument, and then one in Pellicer Creek off of the Favor Dyke State Park boat ramp. San Sebastian and Fort Matanzas are closest to the 2 inlets in our region, the San Sebastian and the Matanzas. As such, they typically tend to be the most influenced by those inlets. Pine Island and Pellicer Creek, however, are much further from the inlets and tend to be more influenced by what's going on in the watershed. And here's a figure of the salinity data, so how salty the water is at all 4 of our sites for the last 19 years. And you can see that the boxes, which represent where most of the data lies, for San Sebastian and Fort Matanzas are much smaller and are higher than those of Pine Island and Pellicer Creek, suggesting very little variability in the salinity at those sites and much more at Pine Island and Pellicer Creek. Again, these sites are much closer to the inlets and are heavily influenced by them. Here's a line actually representing the salinity of ocean water. And so you can actually see how close those 2 sites are to being oceanic. And just on another note, when breaking our data into our our wet and dry seasons of the year, there's actually very little change between those 2 seasons, suggesting that at least when it comes to salinity, spatial variability is likely more important than temporal variability. Essentially, where you are is more important than when you are. And fluorescent dissolved organic matter, I prefer the acronym FDOM for this one, also supports that Pine Island and Pellicer Creek are more watershed influenced, as their FDOM values are much higher than San Sebastian and Fort Matanzas. Essentially, the higher the FDOM, the more color there is in the water, and these sites are often very tea colored. This data is especially exciting because we've only had FDOM sensors on our continuous stations for the past few years. Pine Island and Pellicer Creek are also the ones that are most prone to low dissolved oxygen events. Here are box plots of daily average dissolved oxygen for every single month over the past 19 years. I've marked 2 milligrams per liter with a red line that represents the hypoxia threshold. Essentially, anything below this line is super low oxygen and things like fish struggle to breathe. Notice that there are values that fall below this line at both sites, but particularly at Pellicer Creek. For the daily average to fall below this line, that means that low dissolved oxygen persists in these systems for quite a bit during the day. Pellicer Creek, as Nikki has mentioned, is located within a water body that is impaired by the state of Florida for dissolved oxygen. And so we do typically see low dissolved oxygen values in Pellicer Creek, particularly at the end of the summer and into the fall. Warmer weather, summer rains, and the shallower creek water contributes to the likelihood of the low dissolved oxygen. And over the past 5 years, we can actually see this frequency, especially, but it does look like, at least this past year, it wasn't, we didn't have as extensive an event. That 2 milligrams per liter is the line between the fair and poor categories in this figure. So now we're going to really focus on 3 related parameters. Nikki kind of set the foundation for this in her presentation and that Cloern et al. 2020 paper. So algae are like little plants and they contain chlorophyll A, a photopigment which is used in the process of photosynthesis. So we use this as a proxy for algal biomass. Essentially, more chlorophyll A means more little plants in your water sample. And since nitrogen and phosphorus are critical determinants of plant growth and productivity, we're also going to look at total nitrogen and total phosphorus. What's also been really cool is that since 2018, we're actually able to further look into the breakdown of nitrogen the breakdown of nitrogen into dissolved organic nitrogen, dissolved inorganic nitrogen, and particulate nitrogen. So I'll talk a little bit about that as well. All right, so when it comes to chlorophyll, the punchline is we saw high chlorophyll at all sites this year, especially in the summer months. These 2 box plots are a little different than the ones I've showed you previously. Once again, we have months along the x-axis. However, the boxes actually now contain all of the data through 2020. And I've overlaid last year's 2021 data with a red dot. And I've also included the state threshold for chlorophyll as a dashed horizontal line. Though they're different at both sites, they're both around about 4 micrograms per liter. So San Sebastian on the left and Pellicer Creek on the right have had quite high values for most of the year last year. And San Sebastian actually had record levels in May and in July. I do also want to point out the scale of the axes, the values between the 2 sites. In fact, Pellicer Creek is almost twice the scale of San Sebastian. When the state conducts their assessments of Florida water bodies, many of the criteria are based upon an annual geometric mean. And so looking at the annual geometric means for both of these 2 sites, we do see that the sites quite often exceed the state threshold over the past 19 years. Again, both of these sites are located within water bodies that are impaired for chlorophyll. When looking at total nitrogen, all the sites had high total nitrogen in the summer months, which is to be expected when we get summer rain events. However, total nitrogen was high from June through October at Pine Island, which was much longer than any of the other swamp sites. This figure again shows the annual geometric mean calculations, but this time for total nitrogen. Pine Island is also the only one of our swamp stations where the annual geometric means exceed the state threshold of any of the swamp stations. And this site is located within a water body impaired for nitrogen. And when we break down the different chemical forms of nitrogen, of the total nitrogen, we do see a big pattern that most of the total nitrogen is in dissolved forms, particularly dissolved organic nitrogen. There's a lot of orange on this slide. Also, just a reminder, this is only data from the past 3 years. And our data does differ from that of other estuaries, such as the San Francisco Bay estuary in that Cloern 2020 paper. San Francisco is a well-known system with its own eutrophication story, and it also contains primarily dissolved nitrogen, but it's primarily dissolved inorganic nitrogen. And then when we switch to total phosphorus, it actually seems kind of boring compared to the other two parameters we discussed, because we don't typically tend to see a lot of high phosphorus in the swamp stations, especially not anything to have like annual geometric means exceeding some threshold. Now I'm going to switch to some preliminary work where we're looking into indicators of nutrient enrichment within our system using swamp data. So nitrogen and phosphorus are the elements considered most limiting to plant growth and productivity because they are often present in small quantities or are present in a form that can't be used by a plant. So the adjustment of concentrations of these limiting nutrients can have the effect of modifying a population's size. So greater levels can allow for populations to grow, and whereas reduced amounts should have the opposite effect. So in the early 1930s, an American oceanographer by the name of Albert Redfield examined water samples collected from major oceans around the world and found that no matter where the sample was from, there seemed to be a standard relative quantity of carbon, nitrogen, and phosphorus. This quote and figure are actually excerpts from his 1934 paper in which he postulated that these proportions must be those that are required for the animals and plants that live in the sea. I love that, so philosophical. The ratio has since been called the Redfield Ratio, and it consists of 106 atoms of carbon to 16 atoms of nitrogen to 1 atom of phosphorus. This ratio is particularly useful in determining which nutrients are limiting in a localized system if there is a limiting nutrient. So here we're going to look at those ratios with our swamp data, these nitrogen to phosphorus ratios, but on a biomass scale, not an atomic one. So it transforms our 16:1 ratio to 7.2:1. So I'm calling this the global mean plankton biomass, and it's represented by that line. Anything above this line has higher nitrogen and it has the potential for phosphorus limitation. And anything below this line has more phosphorus and the potential for nitrogen limitation. So here's all of our swamp data, and you can see there's a lot of variability going on. Each point represents 1 month since 2002 at each of the 4 water quality stations. The most variable does appear to be Pellicer Creek. And although there's a lot of things going on, there do seem to be more points above this line, which implies a potential for phosphorus limitation within our system. And here are average nitrogen and phosphorus ratios of all of our swamp sites alongside that global mass of 7.2. As you can see, all of our sites are on average higher than that global ratio. And in fact, Dr. Dix published a paper back in 2013 where she looked at early swamp data and calculated these nitrogen to phosphorus ratios at San Sebastian and Fort Matanzas. And our 19-year average at these same sites are much higher than what she found. And then when we look at that Cloerne paper, they actually found in San Francisco Bay a much lower nitrogen to phosphorus ratio, suggesting that San Francisco is more nitrogen limited. So again, how's the water? We do get it, as Dr. Dick said, quite often. So what does all of this mean? Well, our swamp data does mirror those found or state assessment information. We see high chlorophyll at Pellicer Creek and San Sebastian. We see low dissolved oxygen at Pellicer Creek, though luckily not as frequent in 2001. And then we do see high total nitrogen at Pine Island. Again, each one of these sites is impaired for these same parameters. The majority of the total nitrogen in the GTM estuary is dissolved organic nitrogen, which is kind of cool because there was actually a study back in 2004 at North Inlet Winyah Bay, which is a sister reserve of ours in so many different ways. And they actually found that the high DON, they attributed the high DON to salt marsh, which kind of makes us wonder when we look at these Chesapeake Bay and San Francisco Bay systems that the ratio of salt marsh to water is probably playing more of a role in our system.
1 minute.
And then lastly, indicators of nutrient enrichment. We are seeing these high nitrogen to phosphorus ratios suggesting possible phosphorus limitation, highlighting the importance of keeping an eye on the phosphorus in our system. And the system-specific Redfield ratio could also serve as a proxy for plankton community structure, as this ratio can differ based on the species that are present. So really moving forward, we do want to continue to look at these chemical forms of nutrients, particularly kind of breaking down even the phosphorus levels, We want to link our swamp data with the plankton monitoring that's going on. And then, as Nikki mentioned, we're celebrating 20 years of swamp data here shortly. So that'll be really exciting. And then really quick, lastly, before we open it up to questions, is I really want to highlight the value of the place-based aspect of our swamp research within this national program. We do collect a lot of data, and there's things called tier 1 and tier 2 parameters. And so when you go on the centralized data management office and you look at the parameters that we're all required to submit, you'll see under the nutrients that there are 6 parameters that every reserve is required to collect. But then there's this little asterisk that says, hey, there's also optional things that you could download as well. And so when you click on all the available data for the GTM, you'll actually see we collect a lot of data, particularly nutrient data, that we're not necessarily required to collect. And so the SWAMP program, though it's this national program, there's a lot of more place-based, local, and state initiatives and driven questions that we have, so that affects the data that we're collecting. So if you have any questions, if you want any data, please reach out to any of us on the SWAMP team, myself, Katie Petrinick as our SWAMP data specialist, Silas Tanner, who's presenting later today, as our SWAMP technician, and of course, Dr. Nikki Dix or myself. So thank you so much for listening to me. It's really hard to talk about 19 years of data and find some story in all of it. So thank you so much. And I'll give it right back to you, Caitlin.
Great. Thank you so much, Shannon. I think you did a wonderful job. I know 19 years of data is a lot to summarize, and there have been many potential stories that have come up, especially as storm events happen. So I think you did a wonderful job. We do have a couple of questions in the chat box, and it looks like Dr. Dix is answering them as well, but you might have a different response to some of these. Do you see any correlation between the time or shortly after when thresholds are exceeded and when dredging events along the intracoastal or inlet occur?
We haven't actually looked at any type of timeline associated with that, but that is something, the benefit of having this continuous collection of data is that we can go back and tie events into our data. So that is totally something that we could certainly do. We just don't have the that information at the moment.
Great, thank you. And when it comes to dissolved organic nitrogen, what does that consist of? What is the source, and can it be biologically available?
That is such a great question. And those are actually questions that I've been asking myself as well. I'm more of a fish ecologist by trade, and then I just entered this water quality realm. And so that is a question that I have myself that I'm looking into. Dr. Dix and some of our salt marsh ecologists on here could also probably help address that question. But I really look forward to being able to better understand that and have a really good story with all of this once I do.
And after scrolling down, it does look like Dr. Dix did respond to that as a dissolved organic nitrogen. It's mostly from decaying vegetation, and it can be biologically made. So it just affects different plankton species compositions in the food web. Do you think the high dissolved organic nitrogen could be coming from coastal erosion?
Correlation and causation, I'm not quite sure. There are seasonal depositions of rack and decaying matter that happens all the time. And I say all the time as it's an annual cycle. So I'm not sure where the fine line between what's, I guess I don't want to say normal. I mean, what is normal? But what is something that does happen every year versus things that, like the coastal erosion, things that could be getting progressively more intense. I'm not sure.
And last question in the chat box. Is your Pine Island study site the same one that's near the Nocatee paddle launch? you give a little bit more information on as far as where Pine Island is?
Yes, so Pine Island is attached to channel marker 25 within the Tolomato River. And so it is on the eastern side, the eastern channel markers. And it is, yes, just, I don't know, Silas may know better, but I would say maybe anywhere from a quarter to half a mile downstream from that kayak launch that they just put in Nocatee. Yes.
All right, and we're going to take one last question because I think it's a good one, and then we'll have to stop questions for right now. For the last question, in the 19 years of your data collection, what would you consider as the biggest trend or the biggest takeaway?
That one's hard because there's so many. It really depends on where you are and which site you're interested in. There's global things that we're seeing, warmer temperatures, and what's probably the most funny to me, and I say funny with a lowercase f, is how interesting it is when you do trend analysis with 15 years of data, with 16 years of data, with 19 years of data, how different just adding one more year can really be. And so it seems like we think that we have an idea of something that's going on one year, and then we add a little bit more data, and then it's like, oh, wait, no, not quite. And so we haven't fully done some of our compilation, our swamp status report, if you will, for the full 19-year data set. We're working on that right now. So we'll hope to find at least currently what our status and trends are at present. But again, it is always really funny to me how just adding a little bit more data will change it. So that doesn't help at all, I know. But hopefully we'll have something soon.
That's a great response. Thank you, Shannon.